Hi everyone, and welcome to today's lecture where we will first focus on biotechnology and some ways that we use genes and DNA in the research field, as well as, the medical field and various other areas. And then we'll go more into the gene aspect of development, and then how that will then tie into evolution, which we're going to then cover in the later lectures. So now that you're all experts in terms of DNA, the parts of DNA, the various processes such as replication, transcription, translation, all of that fun, now we're going to look to see, well, what exactly can we do with that DNA? So the first thing I want you to know is what it means when you hear the term recombinant DNA, okay, or recombinant DNA technology. So basically what you're doing with recombinant DNA technology is you are copying, cloning, or manipulating DNA, because recombinant DNA usually refers to DNA that has two different pieces of DNA put together. Okay, so recombinant DNA, think of it as a hybrid of different DNA pieces. Okay, so it has new genes that have been introduced to it. Okay, now when you do recombinant DNA work, meaning genetic engineering, you're going to use two key tools. The first one is restriction enzymes. Those you usually hear called nucleases. Okay, so make sure you write down the word nucleases to describe enzymes that will cut DNA. Okay, and these basically they're highly specific and will only cut certain sequences. And if that particular sequence is only found in the DNA sequence once, okay, you get one cut. If it's found 3, 4, 5 times even, the enzyme will cut every single time that it encounters that particular sequence. Okay. Now, in addition to using these nucleases, you also will use a cloning vector. So you've used those in lab classes while at Georgian Court. Some of them may have been P-Blue or the puck one used in the ligation and transformation kit. Okay, cloning vector is basically a fancy way of saying a plasmid. Okay, so you see the plasmid here, round piece of DNA. Now remember, plasmids are usually these circular pieces of DNA. They're extra chromosomal, meaning that they are beyond the life function chromosomes. And most importantly, these cloning vectors can give bacteria an advantage. And make sure you always know what advantage is it that a plasmid or cloning vector can give to bacteria? Antibiotic resistance. Okay, make sure you write down that cloning vectors can give bacteria antibiotic resistance. So when you're going to use recombinant DNA technology, or basically genetic engineering, a lot of times we call it molecular cloning. And what you have to do is you cut a target gene out of a genome, and then you cut a vector with the same nuclease, the same enzyme. Okay, and once you do that, those two pieces of DNA, your target gene that you cut out of a genome from either human or yeast or bacteria, that target gene that you cut out of a genome now has the same sticky ends as the vector that you just cut open with the same enzyme. So now what you're able to do, as you see in this figure on the slide, is you can ligate them together. Okay, you take that gene that you cut out of a big genome, and you take that vector that you cut open in just one spot, and you glue together those pieces. Once you have that DNA ligated, what can you then do Well, now you could do transformation. And what's transformation? That's basically getting bacteria to take in that new plasmid, that vector, that has your target gene in it. And in the example on this slide, the picture, the target gene that was used is GFP, or green fluorescent protein. And so now that bacteria that has the plasmid with the GFP that you put into it, well, now they can glow green in the presence of UV light. So that's what I said in lab courses when you got to do transformations. It's a very powerful tool. You can give bacteria new genes, which gives them new abilities. They can produce completely new enzymes or proteins that they never had the ability to produce before. And like I said before, remember that one of those things that you can give them is antibiotic resistance, okay, which is very beneficial to the bacteria, and it's also useful in your research as well to help you find the successful transformants later. Now, this is something that you may have seen or remembered from lab, okay? When we talk about cloning vectors, there are 4 main qualities that a cloning vector or plasmid should have in order to be valuable for genetic engineering or transformation, okay? The first one is that the plasmid should be fairly small. And when you ask yourself, why should this plasmid be fairly small? Well, think about what you want to do with it. You want that DNA ring to get into bacteria. And remember, those bacteria have teeny tiny little pores on the outside of their outer membranes. So if that plasmid is nice and small, that increases your chance of getting the bacteria to get that DNA inside of it. Okay, so for the first why should the plasmid be fairly small? To help it get into the bacteria. The next bullet point we have is that the plasmid should have an origin of replication. Why is that? Well, it's in the name. You want that DNA to be copied and maintained in the bacteria once the DNA has gotten in. Okay, so the origin of replication allows the bacteria to copy and maintain your plasmid genes. Okay, then we say that the cloning vector should have at least one selectable marker. Okay, and think of the labs that you've done with transformations. What are some selectable markers that we tend to use? Well, one I've mentioned repeatedly already in the few minutes of this lecture, which is antibiotic resistance. The other is a color change, okay? So if at the end of this experiment, after incubating, your bacteria now can grow in the presence of something like ampicillin, or if it suddenly turns blue when it grows in the presence of something like excal, well then you know those bacteria successfully took in your plasmid, okay? So that's why selectable markers are so valuable, is that they allow you to see which of your bacteria successfully took in the plasmid, okay? Which are the successful transformants. The last of the bullet points is the word that students always hate. They always forget how to say it. That's endonucleases. And endonucleases, what they do is they cut open DNA. So if you remember when we talk about molecular cloning, we said that you want to be able to cut this plasmid open, put new genes into it. Okay, so that's the why of restriction sites or endonuclease sites, is you need to be able to manipulate that plasmid. Okay, you need to be able to cut it open. And usually we call these MCS, which stands for multiple cloning sites. Okay, multiple cloning sites, because these are sites on the plasmid that allow for molecular cloning. They allow you to cut open the plasmid and then insert new genes. Okay, So I'm going to have a brief recap on the next slide just so you get time to write down what we just said. Okay, so here is the recap slide. You can pause the video and take down the notes in case you missed anything I said in the last slide. Okay, so now when we talk about plasmids, a lot of times you'll hear them called multiple copy or multi-copy plasmid vectors. Basically that means, so I'm going to bring all the bullet points out here. Basically that means that these plasmids, especially if they have their proper origin of replication, they can make multiple copies of themselves within the bacteria so that the bacteria can then have high levels of them, okay, which is very valuable to us as researchers because basically the bacteria can become little factories for us, okay, because if they have multi-copy plasmids, they can make many, many copies of the plasmid and then express those new genes at high levels for us. Okay, now one of the most commonly used ones today, it came from what we call the CoL-E1 plasmid that E. coli naturally had. And what researchers did was they took this original, so if you look on the slide here, they took this original plasmid that they found in E. coli and they've removed what was called the colicin production genes. And the reason they removed that is colicin is a bacteriocin. And what that means is it's a toxin that kills some bacteria. Okay, and you may ask yourself, well, why would a bacteria produce a toxin that kills bacteria? Well, it allows it to reduce competition. So especially if they kill off the more similar kind of slightly related bacteria, they get to hoard or have more of the resources they need. Okay, but you don't want that in your plasmid because then it would kill off your experiment. So researchers removed the colicin gene, and then they added what's called the beta-lactamase gene. Okay, they added antibiotic resistance. And why would they want to add the beta-lactamase gene? Well, now if you use this plasmid in research, if it gets into the bacteria, then you'll know because that bacteria will now be able to survive on ampicillin plates. Okay, so you've done this kind of transformation in multiple of your classes, most likely by now. Okay, so keep in mind when you think about plasmid vectors, remember that the original one was from E. coli, and they had to remove the colicin gene and add in the beta-lactamase gene. Okay, and understand why they wanted that beta-lactamase gene in there. So now we get to inserting genes into vectors, which we've, basically been talking about so far, but I want to kind of give you some general rules about doing this. So what you want to do in molecular cloning is you want to cut that plasmid and then cut the gene of interest with the same restriction enzyme. Okay, because we remember we said you're getting that gene of interest from another cell's genome. So you have to cut out that gene of interest, and you want to use the same restriction enzyme so that it will have sticky ends that properly ligate to the vector that you cut. Okay? Now, when you're going to do this, there's certain rules you have to keep in mind. The first one is when you cut, you should only cut the vector once. Okay, you want to open up the vector so that you can glue in the new gene. But what would happen if you cut the vector more than once? You didn't open it up now. You completely chopped it up and it becomes useless at that point. Okay, so you want to make sure you only cut the vector once. You also want to make sure you're not cutting into any genes that are necessary for plasmid stability or survival. If you cut an important gene, then you might end up killing the bacteria when you try to put that plasmid in, or the plasmid won't work. Basically, to help avoid problems, a lot of the vectors of the plasmids have been modified, so they contain what's called a polylinker, or the multiple cloning site we just mentioned a minute ago. That way you have a lot of different options, okay? You have multiple sites to choose from so that you're most likely not, kind of running into the problems we just mentioned. Now, once you have cut the vector and the gene of interest, the next thing you have to do is use DNA ligase to join the ends. So make sure you remember, if you're going to do genetic engineering, you have to cut the plasmid and you cut the gene of interest with the same enzyme. You only cut that plasmid once, and then you use DNA ligase, which is like glue, to stick together the gene of interest and the vector. Okay, so now you should have a nice full plasmid with an extra gene in it. This slide here is to show you what I meant by the polylinker and by the process that we just talked about. Okay, so it helps you visualize. Now, keep in mind, not all plasmids will have that insert or the polylinker, and ligation is not always perfect. Okay, when you do this process, not every vector is going to get cut open. Not every vector is going to get the intended gene, because remember, in lab we had mentioned that when you do this type of experiment, when you cut that vector open and then you put it with these DNA pieces that you want to stick into there, well, look at these. These guys also want to stick back to each other. They match. So a lot of times when you do the experiment, a lot of your plasmids will actually religate on each other and not have the insert. So basically, you need to make sure that there are ways for you to detect which plasmid successfully got cut open and actually took in the gene of interest, okay, which you see here with the plasmid and the gene of interest, rather than just sticking to themselves. So now on this slide, we're going to start going into some ways that you can detect whether your plasmid successfully took in the gene of interest. Now keep in mind, this is different from the selectable markers we talked about earlier. Things like color detection and antibiotic resistance, that was just to show if your plasmid has made it into the bacteria. And that doesn't tell you whether or not your gene made it into the plasmid, because remember, some of the plasmids will just stick to themselves, okay, rather than taking in the gene. So to try and determine whether your plasmid has successfully taken in the gene, you can do a few things. One thing that you can do is you can isolate from the bacteria, pull out the plasmid DNA, and you cut it with the same enzymes that you used in cloning. And think about it. If you look at this picture on the bottom of the slide, if you isolate plasmid DNA from the bacteria and you cut it with the enzymes from cloning, if that plasmid only religated to itself, which is this guy here, then when you cut it with that enzyme, it'll only cut once, and you'll see one linear strip of DNA. If, however, it took in the insert successfully, when you now cut it with the enzyme, look, that's 2 cut sites, right? So now when you run this on a gel, you'll see 2 pieces of DNA instead of 1 linear piece. Okay? If you have trouble visualizing that, just send me a remind message and I'll go over it in a little more detail. Okay, so what we just covered here, if you want to detect an insert in the plasmid, one way is to just simply extract the plasmid DNA and cut it with the same enzyme. If you see 1 band of DNA, then it is linear. It did not take in the insert. If you see two bands of DNA, then it did take in the insert. It successfully took in the insert. Another thing that you can do is insertional inactivation, which is what you did in laboratory if you perform the blue and white ligation transformation technique. And so we're going to go over that one in more detail in the coming slides. And then you have the option to use reporter genes, which kind of a lot of times we combine with insertional inactivation because the reporter genes basically let you visualize things. So these are things like color changes, like seeing blue color of colonies or white color of colonies. So this is the insertional inactivation method of trying to see if your bacteria successfully took in the insert or the new gene. Okay, this is basically what you see in the blue and white ligation transformation experiment. So you have this plasmid over here. Okay, in this example, it's not the blue and white case. In this example, the plasmid will have two different antibiotic resistance genes. Okay, this top one is antibiotic resistance for ampicillin, for instance. This bottom one is antibiotic resistance for kanamycin, for instance. Okay, so AMP resistance and CAN resistance. Now, if you successfully cut open this vector and insert a gene, well, what do you notice? That gene just destroyed the kanamycin resistance. Okay, so any bacteria that has a plasmid that simply religated on itself and never took in a gene, they'll still have AMP and kanamycin resistance. If, however, it took in the new gene, it'll have AMP still working, but it will not be kanamycin resistant. Okay, so in that case, what you would basically do is you would stamp the colonies that you got from this experiment on two different plates, because we have the ability to just stamp them, meaning you put a velvet cloth, you pick up your colonies from your transformation, and you gently touch it on an ampicillin plate, and you gently touch it on a kanamycin plate. And you look to see which of those colonies from your transformation plate successfully grow on ampicillin, but then die off in kanamycin. And you still have the original transformation plate to go back and take those colonies and grow them and use them for your experiment because they have the intended gene. Again, if you're having any trouble understanding this one, just send me a remind message and I'll draw out a picture for you to help you visualize this. Okay, now in the case of our lab experiment, what we do instead of having two antibiotic resistant genes, you have the idea that the vector has the first part of the beta-galactosidase gene, and then the bacterial strain has the second part. So when the regular vector meets the bacteria, boom, you get the blue colonies. Remember, we drew this out on the board. Now, if you then insert a gene into that vector, that beta-galactosidase gene is no longer functional, which means you can no longer get blue colonies. Instead, the white colonies are your success. Okay? Again, if you have any trouble, just send me a reminder message and I'll explain further. Now this one here is basically what we were just talking about, just to give you a visual idea. This is the idea of the plasmid, so just like you did in your experiment, the plasmid having the first part of the beta-galactosidase gene, the bacterial chromosome having the second part. You put the plasmid into the bacteria, boom, you have active beta-galactosidase that produces blue colonies. If you then insert your gene right smack in the middle of here in the plasmid, well, then you know that was successful when you no longer get beta-galactosidase working and you get the white colonies. Okay, so this we went over in lab as well. So this should be basically a review. Now, in addition to plasmid, sometimes we use some other types of vectors. Here we have bacteriophage lambda vectors. That's basically saying that you can use lambda, which is a virus, okay? A virus that infects bacteria with its viral genes. And interestingly, the viral genome of lambda is not actually all essential. So there's a portion of it that you're able to just, like you see in this figure, you're able to just cut out part of that genome and glue in whatever genes you want and put it back into the bacteriophage, into the virus. And now that virus will go and inject that DNA, including the gene you added, into bacteria. Okay, so this is just an alternate route that sometimes researchers use. And when you do this, when you have a viral vector introducing DNA into bacteria, we call that transduction. Okay, so in lab you perform transformation. This here is transduction. Now, another type of research vector that is commonly used is what we call the TI plasmid. This one's used for plant engineering. TI stands for tumor inducing, okay, not a famous musician. So it is tumor inducing, and the reason it's called that is because it's actually naturally carried by what's called Agrobacterium tumefaciens. And you have all probably seen this at one point or another. That's actually a type of bacteria that gets into plants and especially trees through cuts in their, in their bark. So we call that wounds. So for instance, if your parents ever yelled at you, if you're, basically carving into a tree or picking away, peeling with your finger pieces of the bark, when you wound that tree, you open it up to this bacteria. And that bacteria carries a plasmid that has what we call tDNA. And the tDNA, or tumor DNA, basically what that does is what it says, it's tumor causing. It integrates into the plant genome. And what that tDNA has is auxin genes, cytokinin genes, and opine genes. And these are genes that produce enzymes that, as you can see from down here in the figure, promote cell growth, cell division, and feed that bacteria. Now, when you think about this bacteria is putting genes that give enzymes to produce more cells, more plant cell division. What is that? That's cancer, right? That is plant cancer. And that's when you see those giant bulbous tumors sticking out of trees that you're going to see on the next slide. So anytime you're walking by or driving by any of the trees, even on campus, that have these big growths sticking out of them, I want you to shout Agrobacterium tumefaciens, okay? Just shout at it. Look at it. You see, look at that tumefaciens. And all your friends will, of course, think you are the coolest person ever because, that is awesomely cool, of course. Okay? Now, we can then use these plasmids in genetic engineering because if this bacteria has the plasmid which mediates transfer of DNA segments into plant cells, well, then what we can do is we can cut out those genes for the tumor induction and instead put our own genes in there. And the rest of that plasmid is very good at integrating into plant genomes. Okay, so we're able to modify that plasmid, the TI plasmid, to take out the bad bacterial genes and put in our desired genes, and boom, now we can get the plants to produce whatever we want. Okay, so to help you visualize what I was just talking about a minute ago, this is that, crown gall tumors that you might see on campus or various places when you're walking around or driving around. So remember to shout, look at that tumor fascians, okay? And this up here demonstrates what we were just talking about in terms of using the TI plasmid for genetic engineering. So you're able to cut it open, remove those tumor-inducing genes, and ligate in the genes of your desire. Then when you introduce this plasmid into plant cells, well, that plasmid, as you can see here, is naturally very good at integrating into integrating into plant cells and into plant DNA. So now the plants produced from that cell will have your desired genes. So for instance, those genes, think about what might you want to give to a plant. What kind of traits? Well, maybe it's a gene to produce bigger fruit. Maybe it's a gene to produce more fruit, more vegetables, okay, more product. Maybe it's a gene to grow taller, grow bigger. Okay, any of these things resist pests, such as, bugs that might try and attack those plants. So that's how we do genetic engineering a lot of times when it comes to plants and crops, is we use the TI plasmid. Okay, make sure you know if you see a crown gall tumor, though, that's thanks to Agrobacterium tumor fascians. Okay, remember that, it may pop up again. Now, when we talk about genetic engineering, all of this that we're talking about is being able to cut and manipulate DNA. So remember we said nucleases are the enzymes that we use to cut DNA. And there are exonucleases and endonucleases. So if you think of what does exo mean, well, that means outside, right? So exonucleases will cut at the end of a sequence, okay? Whereas endonucleases will cut in the center, okay, within the sequence. So now, with these exo and endonucleases, keep in mind earlier I said that each of these nucleases will recognize a particular sequence, which is usually a palindromic sequence. And it will only cut at that particular sequence. And each enzyme will have a different sequence a lot of times. And like I said, if that sequence is only once in the DNA, then that enzyme will only cut once. If it's 5 times in that DNA, the enzyme will cut every single time it sees that sequence. Now, there are 2 terms I want you to know the difference between. So circle, star, highlight these because these are 2 of my favorite words in the English language. We have isoschismers and we have neoschismers. And both of these are terms for nucleases, but how they differ. Okay, the trick that I use to remember which is which, I think of neoschismers first, because whenever you hear neo means new, okay? So new means that neoschismers are 2 enzymes that will recognize the same sequence of DNA, but will cut at different spots. Okay, so they recognize the same palindrome, but will cut at different spots. For instance, one may cut here, so look over here, one may cut the sequence after the G, whereas the other enzyme, the neoschismer of that first one, will cut after the A or after that A. So they both recognize the same sequence, GAATTC, but they cut at two different places. Okay? Now, isoschizomers, on the other hand, it's a little confusing when I say this. They recognize the same sequence and cut at the same spot. So isoschizomers would both recognize GAATTC, and they would both cut after the G. Okay? That would be isoschizomers. Now you may ask, well, if they're both recognizing the same sequence and they're both cutting at the same spot, why did they have two different names, such as enzyme A and enzyme B? That makes no sense. Well, it makes sense in that they were extracted from different organisms. So let's say you found enzyme A out here on the, soil surface, and you found enzyme B out in an organism in the ocean. Well, yes, they do the same thing, but you name them different things because they're found in different places. And it's so important to know isoschizomer and neoschizomer because, for instance, if you're ever in, medical usage or research field, you run out of a particular enzyme. Well, if you know the isoschizomer or the neoschizomer, you may have access to it, right? Without having to delay your work or run out and order something new. Okay, so make sure you know the difference between isoschizomer and neoschizomer. If you're having trouble, just contact me on Remind and I will give you examples. Again, I will draw them out if you would like. Okay. The other point that I want to make about nucleases is that they'll cut the DNA either leaving a sticky end or a blunt end. And you can see that in this top figure here. So if an enzyme cuts straight down and there's no overhang, as you can see here, that's a blunt end. Whereas if it cuts like this, cuts after that G, cuts after that G, well, now you have that little bit of overhang when they separate. And those are considered sticky ends because think about it. This T is all by itself now, and it's going to want to find an A, okay, to stick to. So that's why they're sticky, because they like to grab a hold of other single strands. And sticky ends tend to work the best for research because, think about it, it's sticky, it wants to bind, okay? The last thing that we have here is modifying enzymes. And modifying enzymes basically means that these enzymes can add a methyl group, which you see in this figure here, and that methyl group actually prevents restriction cuts. Okay, so if you have methyl groups present, it will prevent cutting of that DNA at that particular location. The last thing that we have here is ligases, which you already know that term. Ligases are basically like glue for DNA. They covalently link pieces of DNA, so they're very valuable to manipulating DNA because what good is cutting open that DNA if you don't have the glue to stick back certain pieces together? Okay. Again, if you have any questions about any of these terms we went over on this slide, just send me a remind message. The first thing that I want to mention is in microbiology, we kind of teach you that bacteria aren't always bad. There are a lot of good, helpful ones in your normal flora, and bacteria can be quite valuable and helpful in the realm of research, in the realm of product development. So, the food industry, the medical industry, all over. Some of the ways that they are valuable to us is their ability for bacterial gene transfer, meaning the idea that you could get new DNA, new sequences, new genes put into a bacteria, and you could then have them replicate it, make lots of copies, really play around with the genetic code and get bacteria to be nice little factories for you, basically. Now, there are 3 forms of gene transfer that I want you to know. You have to know the difference between each of them. The first one is transformation. And in transformation, the way we define that is the uptake of naked DNA by bacteria. Okay, so what that means is basically you have a bacteria that needs to be made competent, so meaning that it's able to be, ready to take in DNA. And then you have regular old naked DNA, meaning just strands of DNA around that cell, not in any kind of structure or cell, just free floating DNA, and you get the bacteria to take that DNA in. The other form of gene transfer that you see on this slide that sounds a little similar is transduction. And the difference with transduction is that the bacteria is still taking in new DNA, but the way it's doing it is by virus vector, meaning a virus injects new DNA into the bacteria. The third form of gene transfer that we have here is conjugation, and my students like to remember it as conjugal visits. Okay, if you don't know what that is, I will not explain it, but for those of you who do know what that is, it's a little trick that can help you remember that conjugation is basically a fancy way of saying bacteria sex, because as you can see in this third picture, in conjugation, one bacteria cell is getting DNA from another, and the way they transfer it or they get that DNA is through the formation of a sex pilus, basically a little narrow tunnel for DNA to transfer from the donor to the recipient. Okay, so when you look at these three forms of gene transfer, ask yourself, how is the bacteria getting the new DNA? In transformation, the bacteria is getting pure DNA from the surrounding area. In transduction, the bacteria is getting new DNA from a virus. And lastly, in conjugation, the bacteria is getting new DNA from another bacteria cell. In case you missed any of that, you can pause on this recap and jot down some notes. Okay, so here we have PCR, which, like I just said, is polymerase chain reaction. Its purpose is to amplify DNA, and basically it makes a ton of copies of a very specific piece of DNA, whichever piece of DNA you're targeting. And you may ask yourself, well, why would you want to do that? What scenarios? Well, it's important in things like crime scene investigation, paternity testing, evolution, diagnostic medicine. So you use it a lot in hospital labs as well. Now, in order to perform a PCR, there are various things you need. The first one is the DNA template. So basically the target DNA that you want to amplify. Okay, you need forward and reverse primers, which are little pieces of single-stranded DNA that match the beginning and the end of the DNA sequence that you want to target. Okay, because as you know, polymerases can't start from scratch. They need something to have as starting points. Okay, you'll have the DNA polymerase itself, which is the enzyme that will build the copies of DNA. And it has to be temperature stable, because during this process, you're going to, be using a machine that goes to high temperatures to denature the DNA, so the enzyme has to survive that. You need what are called dNTPs. Those are your nucleotides. So picture those as your A's, your T's, your G's, your C's, the bases that make up DNA. You then need buffers and magnesium chloride, because think about it, you need cofactors for that polymerase. The DNA polymerase is an enzyme, so you need cofactors to help it work, to activate it, and you need to maintain a good environment for this whole process. And the piece that students always forget, you need what's called a PCR machine. You need a very particular machine that will cycle through. It'll go to high temperature, cool down. It'll fluctuate temperatures for you for this process. okay? Now, with that fluctuating temperatures, the reason that is that each PCR, there are 3 steps to happen. You need the machine to first denature the double-stranded DNA, which happens at high temperatures. So you see that here. You heat, the machine will go to a high temperature to separate the double strands of DNA. It will then cool down so that the short primers targeting your specific sequence can bind. Because think about it, if the machine did not cool down, what would happen? These guys would denature too. They wouldn't be able to stick. So it goes from hot to cool, and then it goes to the temperature for Taq polymerase or whichever DNA polymerase you use, because each enzyme has an optimal temperature. Okay, so the first step is high temperature to separate the double strands so that you can then cool the temperature to allow the primers to stick, and then go to the optimal DNA polymerase temperature, such as 72 degrees Celsius, for the polymerase to then build from that primer, okay? And it makes copies of that DNA sequence. Now, you don't have to be able to draw all of this out, but this is just to show you that you don't get your target DNA until 3 cycles later, because each time those primers are, building the polymerase is building from the primers, you end up with a lot of overhang sequences. And as you can see here, this is when you get the nice target DNA. Okay, so you don't have to be able to draw this out or anything. Just remember the fact that it's not until 3 cycles of PCR that you finally get the properly sized target. Okay, circle, star, highlight, 3 cycles. So now I just wanted to show you a sampling of some examples of PCR use, and we're not going to go into the details of, each example and how we actually use them, because that you're going to cover in all of the upper level biology classes. But I just want you to be aware of how powerful PCR is and how it's opened these, endless new research opportunities for us. So we use PCR in genetic engineering, in things like forensic science. So if you think about it, crime scenes, when there's DNA found at a crime scene, it's very small, it's almost degraded. And so PCR allows us to amplify DNA from even the smallest samples of blood or hair and things like that. It allows us to solve crimes in terms of tracing relatives, in terms of rape cases, and even identifying victims where we don't have a body to identify. It's also used in evolutionary biology, basically finding all of the connections in terms of phylogenetics that you see in this figure here. So that's a fancy way of saying, we use PCR to help us trace ancestors and relationships in terms of various organisms. Also used in genetic counseling, which has helped, basically prevent or minimize some of the genetic risks that people may encounter when they're ready to have children with each other. It's used in gene expression research, basically seeing, which genes are upregulated or downregulated in various illnesses or when in the presence of various substances. As you can also see, I listed out the medical field and microbiology, which is a huge area that PCR is used, not just in the development of various medications and drug treatments, but also in diagnostics. A lot of the tests that you take to diagnose or determine whether you have a particular infection or to determine your chances and whether you're high risk for things like cancer. A lot of those techniques rely on some PCR. And notice that I end this slide with "and many more," because to list out the uses for PCR would just be endless at this point, and every day we're coming up with more uses. So just please know these various fields in which we currently use PCR. Now, we just mentioned forensics and crime scene samples using DNA, so I just want to give you a little more information on that. First of all, when you look at forensics, we do a lot of DNA fingerprinting, which is also called DNA profiling. Basically, what you're doing is you determine the genotype of a sample, such as a sample found at a crime scene, or such as a parent sample of DNA, and then you compare it with the DNA profile of other samples, such as various suspects or such as various children. And with this comparison, you then distinguish between samples, and you use this technique to then cancel out who cannot be the criminal or who cannot be the parent in a paternity case. So you're basically using DNA genotypes to narrow down samples in terms of relationships or whether or not someone was at a crime scene. Now, the type of samples that we use, the type of DNA, are called STRs, which stand for short tandem repeats. Okay, the reason we use this is these sequences in your DNA are what we call polymorphic. Okay, basically the idea with this is about over 99.5% of your genome does not vary from the next person. So if you use the majority of DNA sequences to try and distinguish between two people, it's very difficult because most of those sequences, most of that genome are the same. STRs are polymorphic, meaning they have different forms, so they vary between people. So what an STR is it's a non-coding sequence that repeats. They're short tandem repeats. So as you can see here, for instance, one of the STRs is AGAT, and it repeats at a location in your genome a certain number of times. So you would see AGAT, AGAT, AGAT. And each person may have a different number of repeats. Now, when you look at that STR, keep in mind you have two versions of it, two copies, because you got one copy from mom and one copy from dad. So now you could look at that profile and say, okay, the person at the crime scene has at this STR 8 repeats. 8 times in a row, AGAT is repeated in their sequence for one of their alleles, one of their copies that they got from one of their parents. And so if a person does not have 8 repeats in one of their alleles, then you just cancel that person out. They couldn't have been the match for that DNA found at the crime scene. Okay, so a lot of times with DNA fingerprinting, I want you to make note, it does not always tell you who the exact criminal is. Okay, it just narrows down the suspect pool, and then you have to do further sequencing, further tests to make a match. Okay. Now, before we move on from biotechnology, there are just a few terms that I want you to know the definitions of. So first off, I want you to know the difference between genomics versus transcriptomics, and then between transcriptomics versus proteomics. So the words kind of help tell you what they actually mean. Genomics is the study of all of the genes of an organism or a cell, whereas transcriptomics is the study of all of the RNA sequences that that organism or cell can produce. Lastly, proteomics is the study of all of the proteins that that organism or cell can produce. Okay, so you have the study of the genes versus the study of the RNA versus the study of the proteins. And that's important because if you think about it, there are a lot of genes that you have in your cells that are never expressed, that can't produce any RNA or any proteins. So you want to study all three rather than just focusing on one. Now, that then brings us to the idea of systems biology. And systems biology basically allows us to study and understand the interactions between the individual parts of an organism and how that then leads to various properties of that organism. A big example or a big part of systems biology is emergent properties. So if you think about it, emergent properties is basically the idea that some properties you have are the product of a whole bunch of things, okay, rather than just one simple part of organization. So the way we like to kind of explain emergent properties is that the sum of the whole is greater than its individual parts. And what we mean, if sometimes it's easier for you to think of an example to then understand what emergent properties means, is that, for instance, if you're holding a feather, a bird feather in your hand, by holding that feather and looking at it and studying it, you can't explain to me about flight, because being able to fly is a much higher level of organization, a property that develops beyond just the single feather. You would need to know about that whole wing and all the other things that develop beyond that. So a feather, being that small level of organization, does not tell you about wings and the full property of flight, because that's an emergent property that's at a higher level of organization than just that simple feather. So again, when you hear emergent properties, I want you to think of the properties that emerge or come about only when multiple parts interact as a greater whole. Okay? If you have any questions, please contact me in Remind, and I can further explain that to you. Okay, so now we've seen the various ways that we can use genes in terms of research, but now let's look at our own bodies and how they use genes for development. The first thing we have to ask ourselves is, well, what exactly do we mean by the term development? And the textbook definition that we usually give is development includes the processes that allow a multicellular organism to form from an initial single cell. Okay, so development is going from single cell all the way up to the multicellular organism that you are today. Now, with that, there's some terminology I want you to know, because sometimes people accidentally kind of misuse certain terms within development. The first term I want you to know is a zygote, and you see zygote here and also here. What a zygote is, that is the unicellular, so the single cellular fertilized egg. So when an egg becomes fused with a sperm cell during fertilization, that forms the zygote, and it is a single cell. Okay, it's a single cell that now has the complete amount of genes that you are supposed to have. Then what happens is that cell within it starts to split, and you get a whole lot of cell division. This ball now, this ball cell that looks like a whole lot of other cells within it. Then at the point in which this becomes multicellular, that is when it is now an embryo. Okay, so a zygote is the single cell fertilized egg, and then when that eventually becomes multicellular, that is the embryo. Okay, now before it gets there, there's another term that I want you to circle, star, highlight, and that is the blastocyst. And we're going to look on the next slide at a bigger version of the blastocyst. But the reason why this structure is so important is that this is hollowing out here. So this brown gap that you see here, that's hollowed out, and then you have a collection of cells. The cell division has led to this cluster of cells that's called the inner cell mass. And what's important about the blastocyst is that this inner cell mass collection of cells is what becomes the embryo. The outer lining of cells is what becomes the placenta and the other supporting tissues that's ultimately needed for fetal development in the uterus. Okay, so we're going to look at the next slide to focus in on that structure, but please make sure you know the terms development, zygote, embryo, and blastocyst, and then we're going to look at the key points of the blastocyst. So, when you look at the blastocyst, I want you to circle, star, highlight this term here, the trophoblast, because that is the outer lining of cells that I mentioned in the previous slide that becomes the placenta and supporting tissues for fetal development, okay? So circle, star, highlight the term trophoblast, and write the outer cells of the blastocyst that becomes the placenta. Then circle, star, highlight the inner cell mass. This cluster of inner cells is what becomes the embryo. Okay, so make sure you think zygote is the initial single cell fertilized egg. Then you end up with the blastocyst, which has the inner cell mass that will become the embryo, which is multicellular, and the trophoblast, which will become the placenta. Here's just a recap of a few notes that I wanted to make sure that you are able to jot down. So now when we say development, there are 5 essential processes that I want you to know, 5 steps to actually getting proper cellular and organismal development. The first one, which you kind of saw in the zygote picture, the zygote becoming the embryo, is cell division. In development, you have a lot of cell division occurring, and it's very programmed and regulated basically with set times and set locations for this to occur. Then once you have all of those nicely replicated cells, they then start interacting with each other, and you get what's called signal cascades. Basically the idea that there will be signals between various cells to kind of help trigger the next step, which is cell differentiation, okay, which is a process that we'll talk about in a little bit, where these, cells that all look the same, act the same, can suddenly take on specialized functions at particular places and particular times to then form things like specialized tissues and specific organs within the body of the organism. You also then get cell movement that allows for the ultimate, shape that you want in various structures and various tissues and organs. And lastly, to everything there must be an end. And so you also need a very regulated cell death. And I want you to circle star, highlight the fact that programmed cell death is called apoptosis. Okay, whenever you hear apoptosis, that is when a cell is programmed to die, which you can visualize as cell lysis, which is the bursting of a cell. Okay, so in development, you need to have cell division, cell signaling and interactions, differentiation, movement, and ultimately apoptosis. Now, when we talk about in the previous slide, that idea of cell signaling and then cells moving around and taking on new shapes, ultimately this leads to a set body plan. And there are certain terms that I want you to be aware of with regard to an animal's body plan, or what we call the major body axes. Okay, so the first major body axis that I have written here is anterior and posterior. Okay, so whenever you see any of these axes, we write the opposite terms together. So when you look at the anterior versus the posterior of an organism, for anterior, I want you to circle that word and remind yourself, you can kind of associate that with the head structure of that organism. And a little trick that I use to remember what's what in terms of the body axes is when I hear anterior, I think of antlers, because that's a structure that you find on some animals' heads. So the anterior would be the top of the organism, the head area, and then the posterior of that body axis would go down toward their tail. If they have one, with us, it would be the tailbone. But you can think of anterior to posterior as basically top to bottom axis. And again, associate anterior with a head structure. Then you have the ventral dorsal body axis, which is the front of your body. So you can think your abdomen is on your ventral side. And then your dorsal side of that body axis is your back side. Okay, so sometimes you hear with swimming organisms, you hear dorsal fin. So that's the fin on the back side of their body. Okay, so make sure you know ventral is the front side of an animal's body. Dorsal is the back side. And then lastly is the very obvious one that you don't need to kind of memorize any specifics. That's your left to right body axis. Okay, so please remember there are three main body axes, and I'd like you to know the terminology associated with them. Now, when we talk about these body axes, they are set by what's called morphogens. And morphogens are basically molecules that help set up or organize the structure of a body axis. So morphogens will basically help organize where certain structures are found along a body axis. And a big example of a morphogen is bicoid. So when you hear bicoid, I want you to remember two things. First of all, it is an example of a morphogen, meaning a molecule that can set up the body axis arrangement in a developing organism. And secondly, I want you to remember that it is specifically responsible for anterior development. And again, now you're experts on body axes. When you hear anterior, I want you to think the head structure of an organism. Okay, so bicoid ultimately is a molecule that will tell cells where they belong along the anterior-posterior axis, specifically to form anterior structures. Now, a lot of times in the sciences, we actually discover the function of some sort of molecule or hormone or structure based on studying mutants that are called null mutants, meaning N-U-L-L. They completely lack that particular molecule. So the way that they discovered what bicoid does as a morphogen was by studying a mutant that was null. that did not express bicoid. And what they noticed in this example of a fruit fly embryo is that the mutant that does not produce any bicoid suddenly does not have these anterior structures. Notice over here, it lacks the anterior structures, and instead it just looks the same as some posterior structures. So when there's no bicoid, there's no anterior, and that tells you that bicoid is responsible for forming the anterior. So now what I want you to do is to send me in the Remind app right now, okay? Please don't forget to do this. Send me in the Remind app what you think would then happen if a mutant produces twice as much bicoid. So this here shows in the figure what happens when there's no bicoid. What do you think would happen when there is double the amount of bicoid, and why? Now, another way that we kind of help figure out what something does, or kind of where you might find it in a cell, is called FISH. So FISH has helped us study morphogens. FISH stands for fluorescence in situ hybridization. And what this process is first of all, it relies on a small DNA probe. And what that means is when you have a single strand of nucleic acid, like I've said before, single nucleic acids, they're single. They're looking for their partner. They're very clingy. And so they look for their match and they will cling to it. And their match is their complementary sequence. So what we do is when we make this single-stranded probe, we make it match whichever piece or sequence of RNA we want to target. So it will only stick to that target. Then what we also do is we make sure that it has a fluorescent label. And so when that probe is bound to the target mRNA that it matches, you will see fluorescence wherever that is in the cell. So what they did here in this figure, they made the probe to match the bicoid mRNA. So this probe will only bind to bicoid mRNA, and you'll know where it's bound because you'll see fluorescent in that part of the embryo. And it will be green, bright green fluorescence. And when you look at this picture of the Drosophila embryo, what do you notice? You see that bright green fluorescence in the anterior portion of the embryo. So this tells you that the bicoid mRNA is concentrated at the anterior end of the embryo, and that's where it is functioning, okay? Because we said bicoid is for anterior production, right? Anterior structure production, and basically the anterior mapping of what cells go where on the anterior side, okay? So the purpose of FISH is allows us to visualize and find the exact location of a specific target mRNA in an embryo. And in this example, it was of the bicoid mRNA. Now we keep saying morphogens like bicoid, they help map what cells, what structures get formed at particular locations along the axis that we're looking at in the developing embryo. Now the way that they do this is through differential gene expression, which is based on the presence or absence of regulatory transcription factors. So for instance, bicoid, we not only refer to that as a morphogen, but bicoid protein is actually also a regulatory transcription factor. Because, like we said, bicoid triggers the formation of anterior structures, right, in this location. And the way that it does this is through signaling and helping to activate certain genes or repress others. And what you'll notice when I say differential gene expression occurs through the presence or absence of these transcription factors, well, look at the embryo. You get certain genes expressed on the anterior side because of the high presence of bicoid. And then because bicoid decreases toward the posterior side and there is little or no bicoid on that posterior side, you get a completely different set of genes that are expressed in the posterior because of the absence of bicoid. Okay, so the presence of bicoid on the anterior and the absence of bicoid on the posterior basically affects which genes get transcribed, which structures get formed, and then in areas where it's lacking bicoid, then that determines which structures do not get formed, because certain genes are not activated or no longer activated in those locations. Okay, so again, the exact mechanisms we'll go through more in the upper level courses, but for now, please make sure you understand the idea of this gradient of gene expression, allowing for the ultimate axis structures to form. So now we've talked about that differential gene expression and how it's based on these regulatory transcription factors, and ultimately that leads us to what's called the genetic regulatory cascades. And what that means is that you end up with a set of regulatory genes connected in a way that when the first one is activated, that then triggers down the line the activation of a series of other genes to then ultimately lead to the expression and the overall, differentiation and appearance of what the structures and the embryo is supposed to be in development. Now, the way to think of genetic regulatory cascades is basically like a domino effect, a series of dominoes. And the ultimate purpose of this domino effect is that all of these different genes in a row lead to the information that the embryo needs to have certain cells at a certain location. Certain structures need to be produced at certain areas, and we get this through these series of gene activations. Now over here I put from your textbook an example of a genetic regulatory cascade. It starts off with our famous little friend bicoid, the morphogen that we mentioned before for anterior versus posterior development. And so these morphogens in the first step will control the formation of main segments of what the embryo should be, so basically determining anterior versus posterior. Then you have what's called gap genes that get expressed, and through the activation from bicoid or other morphogens, these gap genes then control the formation of more specific, smaller groups of segments within the embryo that will define large regions of the final body of that organism. Then there are pair rule genes next. And so as you can see, this is cascading where gap genes are in response to morphogens, and then pair rule genes are in response to the gap genes. And these pair rule genes will ultimately control the formation of individual segments within that embryo, and then that will trigger segment polarity genes. Okay, and these segment polarity genes will basically allow for specific regions to be restricted versus specific regions that need to have expression. Now, what I want you to then circle, star, highlight is the term Hox genes. That's the next step, and Hox genes are the specific one that I really want you to know, because these are genes that ultimately will help regulate and specify each segment of that embryo, its identity. Okay, so Hox genes are very important in developmental biology to ultimately define, what structures get formed. And then lastly, you have the effector genes that are upregulated or activated through the Hox genes, and they will cause the ultimate developmental steps that we mentioned earlier, including cell proliferation, which is a fancy way of saying cell replication, ultimately cell death, which we know as apoptosis, and the various other steps that we talked about. Now, you don't have to memorize all of these steps. The key that I want you to know is what exactly is a genetic regulatory cascade. Why does this occur? What's its purpose? And I want you to know the Hox genes that we're going to talk about in a little more detail in the coming slides. Now, here's just a visual of what we just talked about in the previous slide for you to actually see how you start out with those undifferentiated cells of an embryo where they all look the same, they act the same, they have no specific purpose. And then through this genetic cascade, that domino effect of morphogens activating gap genes, activating parallel genes, then segment polarity, then the Hox genes get activated and the effector genes get activated, and ultimately through all of these different steps of activation, one after the other, you end up with the differentiated cells and the development that you need to become a full-blown organism. Now again, notice the terms that we mentioned earlier come back. So this whole genetic regulatory cascade leads to the cell replication and the cell movement, shape formation, differentiation that you need to become a fully developed organism. Okay, so now with that genetic regulatory cascade, sometimes things go wrong, and that brings us to one of the really cool visuals of this lesson, which is homeosis, also known as homeotic mutations. And so when homeosis occurs due to homeotic mutations, that means that you end up with body parts in the wrong place. And so you see that in the pictures here. Here you have a normal fruit fly, Drosophila, where it has little antennae sticking out from the front of its face. With homeosis, due to a homeotic mutation, now you see, boom, it's got two legs sticking out where the antenna should have been. Then down here, you also have examples of homeotic mutations or homeosis, because here is a normal wild-type fly with just two wings. Then when the homeotic mutation occurs, look at this. You have two wings in a location that they are not supposed to be. So you end up with extra wings. Now, when this happens, ultimately it is a problem with the embryonic messages along the axis of development. And the reason you get these are mutations in particular genes. Now, it varies depending on whether you're looking at an animal homeotic mutation or a plant homeotic mutation. If I ask about animals, then Hox genes are what's mutated with homeosis. And if I ask about plants having, parts in the wrong place, then it's MADS genes. Okay, so either one of these genes, if you have mutations in them, it's called homeotic mutations, and you end up with parts of the animal or parts of the plant in the wrong place. And so this is just a visual because I know you're thinking body parts and you're like, wow, how does that happen with plants? But homeotic mutations can happen in plants. It is in the MADS genes. So when I say then which genes is the mutation occurring in, you write MADS from the previous slide. And an example of this is you look at a regular wild type flower and you see that there's a stamen and you have the carpel right there. In a homeotic mutation, you end up with flower parts in the wrong place. So notice you're supposed to have the sepal and the petals here, and then the stamen and carpel. In the homeotic mutation, where the carpel and stamen should have been, look, you have a whole bunch more petals and sepals where they're not supposed to be. Okay, and so that's due to mutation in the MADS genes, which are the plant equivalent of our animal HOX genes. And any of these genes are responsible for ultimately the proper development or pattern of where body parts are supposed to be or where plant parts are supposed to be through development. Now, with this idea of development and patterns and all this kind of gene regulation, I want you to know two more terms, which are commitment and determination. And I know you live with these terms in everyday life, but specifically with regard to biology and especially developmental bio, commitment is basically when an embryonic cell is going to become dedicated to a particular path of cell specialization, meaning it's going to go from undifferentiated, all the cells looking the same, not being specific, not having an exact purpose. Now it's going to follow a path towards specialization, okay? Now, with this, once a cell goes down this path, okay, it's going to become more and more committed to that ultimate fate or that ultimate specific cell that it's supposed to become. So early on in commitment, it's very easy for the cell to reverse paths and not go down that final stage, pathway of differentiation. The further along the path the cell goes, the stronger the commitment becomes, and the cell ultimately will become locked in to that particular fate. So let's say I'm an undifferentiated cell and I'm going to commit to becoming a cardiac cell, a cell that's part of your heart tissue, okay? In the beginning of that pathway, I could still turn around and not become a cardiac cell. But as I go further along that pathway and you get that cascade of gene regulation that we talked about, eventually I reach a point where I am locked in and I have to become that cardiac cell. And when I am locked into that particular type of cell, that is called determination, okay? Now I am said to be a determined cell, okay? So commitment is technically reversible early on. The difference with determination is you are now locked into that differentiated cell, that exact specific cell that you are going to become, okay? Now along that path from commitment toward determination, you encounter various master regulators. A master regulator is simply a gene or protein that will trigger a series of events producing the specialized cell type or tissue that you are going to become. An example of a master regulator, which is found in the process of when your cells become committed to differentiating into muscle cells, the master regulator there is MyoD. So MyoD is an example of a master regulator involved in triggering undifferentiated cells to become specifically muscle cells. And this here is an example. Steps 1 through 4 are examples of steps in the process of commitment and determination, specifically the path that cells in your bicep or any other muscular area of your body would take to become muscle cells. Okay, and you notice that early on, while in, for instance, the early embryo stages, a lot of the cells are undifferentiated. Okay, they don't have an exact purpose or cell type yet. Then they begin to produce signals that trigger that regulatory gene cascade that we talked about in previous slides. And ultimately, the signals will then trigger the production of a master regulator. In this case, the master regulator being MyoD. And MyoD producing cells will ultimately express and produce the specific proteins that then make up your muscle cells. So for instance, if you're thinking in terms of actin and myosin, okay, you need specific proteins and structures for that differentiated, determined muscle cell. Now that concept of commitment and determination and differentiating cells leads us to the idea of the stem cell theory. And ultimately what stem cells are, sometimes you hear them called induced pluripotent stem cells. These are cells that are undifferentiated, meaning they have not been committed or determined yet. So it's kind of like when they say, well, I could have been anything. Well, these stem cells are at the point where they can be anything. They're little undergrad cells that can choose any path they want and go down that path to ultimately be determined and differentiated. Now, what's cool with the stem cell theory is that scientists have been able to convert some differentiated adult cells, so they've already been determined, into embryo-like cells. Okay, so that's why you come across the term induced pluripotent stem cells, because the scientists have been able to take cells that were already differentiated and induce a stem cell-like nature where they can now become whatever cell you want to differentiate them into. Okay, and that has a lot of potential in terms of the medical field and in terms of treating various illnesses, especially things like neurodegenerative issues like Parkinson's, Alzheimer's, things like that, where you lose a lot of the adult cells that are already differentiated, that are necessary in your body, and you're not replacing them. So induced pluripotent stem cells would be a way to replace them, to turn these undifferentiated cells into the exact cell you need, such as nervous system cells, okay, neurons. So it has a lot of purpose, but there's still a lot of various, obstacles that research is facing, especially with, ethical pushback of people who don't understand the actual process. Okay, so has a lot of potential though. Now, although we have predominantly focused on gene expression and genetics these past few lessons, we're going to be heading into more evolution and ecological studies, and they actually tie very close together. So when we talk about developmental gene expression, like we did in this whole lesson, you have to realize that changes in that developmental gene expression can lead to organisms having different structures, right? Different abilities. And this is very important because if you think about it, differentiation, we said, is caused by, all this differential and regulatory gene expression. And it ultimately triggers these cascades and these processes to develop certain structures. But if one of these processes is now changed, okay, you may end up developing a structure with a different size or a different shape or even yet a different ability. And that's critical for evolution because it creates biodiversity. Okay, biodiversity meaning diversity and variations amongst different organisms. And that's very important in evolution because some of these changes in development might give an advantage that then helps develop further organisms that, have bigger, better abilities, okay? So it causes these cascading changes. And with that comes the emerging field of Evo-Devo. I love that term, Evo-Devo. It's basically looking at evolution and developmental biology. So now it's evolutionary developmental biology, combining the two studies and kind of looking to see how different organisms develop the way that they have and how that affects their abilities, how they interact with each other, how they interact with the environment. A very cool example of Evo-Devo, looking at how developmental gene expression can affect an organism's development and evolution, is the loss of limbs in snakes. Okay, what's cool here is if you look at this figure that explains the loss of limbs in snakes, it combines evolution and development because we talked about in development this lecture, Hox genes. And as you can see here in this chick embryo, when you have just HOX6 somewhere expressed, with no HOX8 being expressed at that same location, you end up with a forelimb, meaning in this case the little wing, or in another organism a leg, an arm, at that location. If, however, both Hox genes, HOX6 and 8, are expressed at the same time, instead of a limb, you get a rib. Now, what do you notice about the snake embryo with development and gene expression? All along that embryo, HOX6 and 8 are expressed together at the same time. There is no location on this embryo where you have just HOX6, which means there's no location where forelimbs would develop, okay? So through this change and evolutionary gene expression of having both Hox genes 6 and 8 all along the embryo of a snake, as opposed to chicks and other organisms where it's varied and you have individual expression and forelimb development, snakes, you notice, have nowhere for forelimbs. Now I want to point out something else interesting with this information. Not only is it that this leads to no forelimbs, what did I say develops instead when you have 6 and 8, HOX6 and 8 expressed at the same time? Instead of a forelimb, you get ribs, right? So when you look at that figure, you have to ask yourself, well, what's different about that snake in evolution compared to other animals? Not only does it not have limbs because there's nowhere with just HOX6, but because HOX6 and 8 are expressed throughout the whole embryo, snakes have between 200 and 400 vertebrae with ribs attached. So snakes have between 200 and 400 ribs because of this differential gene expression, because of the fact that they have HOX6 and 8 all throughout their embryo expressed together, rather than having locations where HOX6 is expressed by itself. Okay, I want you to circle, star, highlight HOX6 and HOX8 genes, and make sure you know that if it's just HOX6, you get a limb. If it's both together, you get ribs instead. And to know the difference between snakes and other organisms and how they end up with so many ribs and no forelimbs. It's because HOX6 and 8 are always expressed together in a snake. Okay, circle, star, highlight that information, please. Okay. And that is it for today. I know a lot of students get bogged down by all the genetic stuff, so at least you have made it through these lessons and will be able to move on toward a few other different topics, get away from the genetics a little bit, but please do not forget the material we covered so far, because the genetics information will come back throughout multiple lessons as well. It will help you understand a lot of the other topics. Plus, it's the professor's favorite topic, genetics. So that is it for today. As always, if you have any questions or concerns, please contact me in the Remind app, day or night. Like I always say, I have no life, so I don't mind.